Welcome to biology. In this video, we are going to review the process of RNA transcription. Now let's remember, the location of RNA transcription happens in the nucleus. And this process involves one single enzyme, and that enzyme is called RNA polymerase. Now, RNA polymerase has a couple jobs, and that first job is to unwind the DNA. Then RNA polymerase attaches the complementary RNA nucleotide base pairs. So on my image here, I'm going to color in the RNA polymerase. So I'm going to pick red for this, for my RNA polymerase. And that is this big enzyme over here. Now I also want to color in the DNA so we can see where that original strand was that is unwound by this RNA polymerase. Now I am going to use green for my DNA, and I'm only going to color in the backbone of the original DNA. Remember, the process of RNA transcription is the process that converts DNA into messenger RNA. So that RNA polymerase first unwinds the DNA, then it attaches the complementary RNA nucleotide base pairs, and at the end, it creates new messenger RNA. On my diagram, I am going to color in that new messenger RNA along the backbone right here. Inside of the RNA polymerase enzyme, we can see these new nucleotide base pairs being brought in to attach in to the newly formed messenger RNA. Now this messenger RNA is going to leave that RNA polymerase after the transcription has reached a stop codon. And when that messenger RNA leaves, it is going to exit the nucleus of the cell and head out to a ribosome. But let's first quick review RNA base pairing. Remember, in RNA, we have our cytosine bind with guanine, and this is the same that we have in DNA, but there's no thymine in RNA. So adenine is going to bind with uracil. So again, remember, there is no thymine in RNA. Again, remember, when cytosine binds with guanine, that creates 3 hydrogen bonds, whereas when adenine binds with uracil, just like when adenine binds with thymine in DNA, we only make 2 hydrogen bonds.